Thanks very much. My first contact with ME came as an ME-associated family, and of course then as a biomedical researcher I was really keen to try and get involved in research and look for an opportunity to join a research program where I thought actually we could make some progress and some contribution. I have to say it's the passion of my personal experience with ME which drives me in this research, but there's a little kind of voice in my head all of the time which reminds me when I get too excited. It says, is this what you're planning to do translatable to actually benefit patients with ME? So I try and keep that grounded as a message to myself that whatever we do, we have to be conscious of that. I have to say I feel a bit empathetic with patients in the audience, and this has been a long day because this is about 4:30 a.m. New Zealand time for me, so I'm feeling very jet lagged and a bit fatigued. So
you want your bed?
I want my bed. Good. So my first contact then was when my 14-year-old daughter, a very vibrant young teenager, a couple of months or so after an Epstein-Barr infection, came down with ME, as we subsequently discovered, and she had very severe symptoms and a wide range of symptoms, and of course I was just amazed at a total energy collapse. Her homeostatic mechanisms were all over the place. One day she would have a blood glucose of 2 and the next day it would be 16. She became allergic to all of the food items she normally consumed, and so my wife had to sort of deconstruct her whole diet and go back to fundamental food items and build up a group of things that she could take without response for sustainable nutrition. And she was a very high-achieving, bright girl, totally intimidated me as her parent at the time, and then amazing effect on her cognitive ability as a result of the illness. So, of course, I asked myself, what major control center in the body is directing all of this, the severity of all of the symptoms and also the diversity of them where so much of the body system seemed to be affected. All my daughter asked of me was, why am I so ill? And then, of course, the illness, didn't get better. And so the question for me is, what perpetuates this illness? And these questions really still, are in the forefront of my mind today as I'm doing the research. Again, in a very humbling way, my daughter just said to me, why am I not getting well? And then she, after about 4 years of an acute illness, she kind of crawled out of that and got into what we'd call the chronic phase, but then any kind of whiff of a stress would put her into relapse, a cycle which she'd have to gradually pull herself up. So the question I asked, what's causing biologically these relapse cycles? And so these are questions which sort of I hold on to. And the good point of the story, as I told the biomedical symposium in the last couple of days, is that 25 years after she onset of the disease, she became pregnant and her symptoms dramatically improved, had the best year of her health in all of the 25 years of the illness. So I was just amazed how the physiological changes, is it the increased blood volume, the suppression of the immune system, this more stable hormone status, which actually sent the illness. But to me, that was a great hope for all sufferers of ME, because it said, if we can tweak some things there, that one might be able to get a vast improvement in the status for sufferers of the illness. And I was very impressed with Olaf's comment that, that when these drugs that he's been giving his patients, that actually all of the symptoms go away together, which is again a reinforcement that actually it is possible to do this. as I was thinking about how I would actually get into the research of this illness, it struck me that actually the paradigm that I'd been using for my biological research wasn't really adequate. And because I was looking at all of the research that had been published at that time, and they were interesting studies, but they were like pieces of a puzzle. And when I looked at those pieces, I wasn't able to actually see what the picture was. I couldn't immediately see what that puzzle was about. And so that said to me that somehow we need to approach studies with this illness in a different way. The traditional way of having a narrow focused hypothesis and then answering, doing research on that hypothesis to get answers had produced these puzzle pieces, but it hadn't given us an idea of the fundamental mechanisms underlying the disease. And so I came to think that I needed to step back and actually collect data from well-characterized patients. And because I'm a molecular biologist, it seemed to me that it would be valuable collecting multiple molecular classes, data on multiple molecular classes to advance the disease. So what I hoped we would do then would be, by doing this, we might be able to get pieces of the puzzle which were a bit more definitive. If you saw them beside other pieces, you could get an idea then what the overall or integrated image was. So essentially I was wanting to solve the puzzle, and I have to say, when I first saw this, I could never tell that this was the overall image that we would have actually got out of it. And that's how I felt about ME, that we needed to somehow get data which could give us that arrow, that direction through to the final picture. Now, I took my inspiration in thinking how I could channel this into a research program from an ex-student, Tony Manning, who works in the biotech industry, and his company is actually working on rare diseases. Here he is here. And because rare diseases have only a few patients, they've used the concept of what we now know as precision medicine, where they've looked at multiple molecular classes and tried to integrate the data from each of these to get an understanding of what the rare disease is about. And they've had one or two quite dramatic successes, particularly in immune disease and also autoimmune disease. And this seemed to me to be a very good model. And so in talking with him and talking about ME/CFS, he said that he could see that ME/CFS was a great disease to apply these principles to. So that was one of the stimulations of where we should go. The other one was a study that came out in the journal Cell in April of 2012 from a Stanford geneticist, Michael Snyder, where he took blood samples over a 2-year period and did every omic study that you could imagine on his blood during that sort of period of time, multi-authors on the paper. Now, the outcome and the data was not so profound, but for me it was that, wow, in terms of doing longitudinal studies on ME patients, this kind of approach could be incredibly valuable. In other words, we could understand what was going on during the relapse recycling study. And there was a question, I think, from this gentleman down here in the last talk, which really asked about this. And so we're starting a study on this now where 2 of the patients in the cohort studies that I'll mention in a moment do have frequent relapses. So I've asked them whether they will give blood every month over a period of time, and we'll try and catch them through relapse and recovery cycles to see if we can see actually what changes are occurring. So I thought that was a very perceptive question. Okay, so the direction forward, the success of the rare disease studies suggested that precision medicine was a very good way to go. And the concept of you can actually gain a lot from looking at a comprehensive molecular history in a longitudinal fashion with each individual is a very powerful tool. The problem is that original study was a $2 million study, and not even in New Zealand do we have $2 million to spend on each patient. But Kiwis are pretty parsimonious, and so I thought about, what kind of classes of molecules should we actually examine so that we could get a tightly budgeted study, but still very valuable. And so that's where we've gone. And so what we're doing then is collecting many puzzle pieces at the same time, in my puzzle analogy, by doing different studies within each patient. And we're trying to assemble them into the picture, and by linking molecules together in functional associations, which will give us insight into the dysfunctional pathways that are occurring in the patients. And then finally trying to start building models. And I think from what you've heard today, actually we're in a very good position now to start thinking about alternative models which might answer some of those big questions that I actually posed at the beginning. So we set up two small preclinical CFS studies. Firstly, the Dunedin study, where I'm from, which was a precision medicine study of 10 patients and 10 controls. The patients were all diagnosed by our ME clinical expert who's in the audience, Dr. Rosamund Vellings, and they were well characterised, so we had sort of good data on them. And the molecular studies I decided to do was cytokines, because clearly as immune modulators, and the immune system is clearly involved in the illness, and also so that we could get data that we could compare with other studies that were emerging in other centres. Decided to also look at plasma microRNA, and for those of you that don't know too much about microRNA, in the last 10 years we've discovered that microRNA are small RNAs that have an incredible, profound effect on controlling all of our physiology. And remarkably, because I'm an RNA biologist, RNA is normally quite unstable, but they're found in plasma in a stable form, and they can reflect what's going on in the body with the healthy or unhealthy physiology in the body. So there's a range of microRNAs in the plasma, and if you have some problem with your liver, then you might get elevation in the plasma of microRNAs that are specific for that. So that was the underlying philosophy and concept that we wanted to go by. And then to take purified immune cells, lymphocytes, and actually do an analysis of all of the expressed genes, so-called transcriptome, and also all of the proteins, so-called proteomes, so we could actually look to see what changes were occurring in the immune cells in this illness. The second cohort, the Palmerston North cohort, also 10 patients and 10 controls, and this was done in collaboration with exercise physiologist Lynette Hodges, because the aim of the study was to look at exercise intolerance, but the before exercise samples, of course, they were able to be a second independent cohort together with our Dunedin cohort. Lynette was amazing. She also recruited 5 MS patients who were prepared to come in and exercise one day and then come back 24 hours later and exercise again. So we not only had our ME CFS patients, but we had MS patients as a control. So far we're in the middle of doing all of the molecular analyses on this study, but we've got cytokine data, and I'll show you that in a minute. So this MEC student, Alex Noble, did the cytokine work, and what we immediately found, again, a feature which is well known, I think, in ME CFS, that cytokines that were involved in acute inflammation like IL-8, and chronic inflammation, IL-7, VEGF, and IL-13 in our case, we identified as being elevated in the chronic fatigue, ME chronic fatigue syndrome patients. And you see just the example of a box plot there showing the data just at the bottom right, an example of the data. PhD student Erin Sweetman looked at the microRNAs, and we used a TACMAN system, card system, examined 781 microRNAs in the plasma, and got 3 that were significantly elevated, or 2 significantly elevated and 1 decreased in plasma 7G, microRNA 142 and 1825. Interestingly enough, 142P has been connected with IL-8 in macrophages during chronic inflammation. So one of the things we wanted to do by doing multiple classes of molecules was to see if we could actually cross the classes of molecules and to get much more informative information. And so here, just in the first 2 examples of these studies, we were able to connect one of the microRNAs that was elevated with one of the cytokines in inflammation. MicroRNA 1825 is kind of like a master microRNA that controls a lot of other ones, suggests that overall in the illness, perhaps a number of microRNAs are being affected. Of course, we were looking at plasma, maybe in cells. There were 3 others that were near to significance. We then went on and then looked at the immune cell transcriptome, and I just want to illustrate this with this piece of data. The top 10 gene transcripts that were elevated, we could show connections, quite a few functional connections between those elevations. And if we took the top 3, one of them, again, was the transcript for IL-8, which we'd identified in the cytokine study, and the next 2 were actually modulators of the production of IL-8. So it looked like there were 2 genes elevated here, which was trying to dampen down the autoinflammation. So functionally, we were starting to generate quite a lot of interesting information just by looking at the top part of the data. This piece of data came from where we just took the ME patients and we compared them with the controls. But we then took a step back and allowed a statistical multivariate analysis to actually separate the data out to see if we could get individual groups where we had the controls and patients blinded. And then we found that there were over 2,200 gene transcripts that were elevated in the ME/CFS patients and 850 that were downregulated. So enormously rich amount of data suggesting there are major changes in the molecular biology of the immune cells. We then went on and did our proteome study, and this is an example of firstly doing this principal component analysis where we have the individuals blinded and allow the analysis to see if we can separate out into 2 groups. And what you'll see here, we had 2 ways of doing this, and I've color-coded this. One way of doing it was to actually take this group and take this group, or alternatively we could just take a line over the 0 and take those below and above. So this is a mass spectrometry swath analysis where we actually do triplicate analyses. So you'll see sort of groupings of 3 spots here, and that's the 3 individual analyses of the one protein. When we took the across the 0 line, in fact, all of the group below that line were all ME/CFS patients. And of course, that meant all the controls were in the other group, but there were also 1 or 2 ME, I think there was 2 MEs patients that actually were in the control group. So this is quite a good way of kind of getting the data sorted, and you get a more rich analysis out of this. So from this, we were able to show that a number of important biochemical pathways were modulated, and you could look at the functional associations between proteins in those pathways, and that's what you're seeing in the right there. And you can see those connected lines suggest that a number of the proteins that were elevated in the pathway were actually functionally connected. And what I've highlighted there, that two of those, one's an inflammatory response, one an acute inflammatory response. So this is a theme which is coming through all of the classes of data. We firstly identify the inflammatory and chronic inflammatory cytokines, connections in the microRNAs with inflammation, and then here it is in the cellular proteome. So if one looks then globally at what we found from the cellular data, all of these thousands of, or 3,000 gene transcripts that were affected, somewhat less of the proteins because the analysis only samples about 1,875 of the proteins. We could say that enriched were pathways involved in the immune system, inflammatory pathways, pathways which would be labelled as cytokine and apoptosis, programmed cell death. And decrease, we could see proteins in the mitochondria involved in energy pathways decreased in amount, and general metabolism, particularly in the transcriptome, a lot of genes involved in different metabolic pathways, and this is consistent with the metabolome data that came out with those beautiful studies last year. And from the proteome, it looked like lipid metabolism was being quite dramatically affected. So we've got a lot of information coming out of that study. I want to just now say a word or two about the exercise intolerance study. Lynette Hodges is an exercise physiologist, so she made all of these measurements here in those parameters. And the study was that the patients came in from day one, and they started at 15 watts power, and then increased that by another 15 watts every minute until they actually couldn't go on any further. We got blood before the test started, and then 24 hours later they came in and we got a second blood sample, and they did a second exercise test, and so we looked at the data, and I just want to show you a little bit of data there on power output. So here what we're looking at are three subgroups. The one in the middle is the healthy controls, the one on the right, the MS patients, and the CFS ME patients on the left. So with the healthy controls, you can see on day one, there's heart rate there. In day two exercise, heart rate goes up a bit more, and their power output increases from day one to day two. And I'm not an expert on this, but I'm told this is not unusual to happen when you do a re-exercise test. Interestingly enough, if we look at the MS patients, their maximum sort of heart rates are lower, but there's a marginal increase from day one to day two, and even for power output, of course it's low on day one, but a marginal increase or no change from day two. But when you come across to the ME/CFS patients, you'll see that they start off on day one, in terms of maximum heart rate, about the same as the healthy controls. It's lower on day two, but when you look at the power output, there's a significant drop from day two to day one, which you don't see in MS and you don't see in the healthy controls. And I guess that's an indication of the exercise intolerance. So what we were interested to see is what happens with all of the molecular classes, and I've just got data on the cytokines, which I'll show. And this is quite a complex slide, but really I want you to just focus on the arrows. So we've got 3 columns of cytokines there, and within each of the columns there's ME patients on the left and there's controls on the right. And you can see if you go down the left-hand column, so which is there's a tendency for the cytokine to go up 24 hours after exercise. There's one exception on the right. But if you look at the controls, the cytokines all go down, and that's what is expected and what goes down. So what we're seeing here is the ME patients aren't responding in the same way to what normally happens with the cytokines. If anything, it's going in the opposite direction. So at least that's some molecular change which relates to the fact that they don't exercise as well on the second day. Okay. One thing that Alex Noble, the MSc student, did of her own volition was to say if she took the top 4 cytokines that changed and she modelled all of the patients that she had examined to see what those 4 most significant would predict. This is the data that came out. So there were 19 controls in the group. There were 14 ME and 4 MS. And so the model predict on the basis of these 4 cytokines, 27 controls, 10 ME and 0 MS. So that doesn't look too good, except if you deconstruct what it really is saying. The controls, 18 controls were scored as controls, 1 came out as ME. For the ME patients, 9 came out as ME and 5 as controls. But we found when we did the exercise parameters that 3 of the people didn't look like ME CFS at all. They just looked like healthy controls. So presumably we're thinking that 3 of those are scoring there. And then the MS people who of course didn't look like an ME patient, they looked more like a control, a very unfit control, I might say, but they've clearly all been scored as controls. So I'm not saying this is we would use as a predictor for the disease, but it's interesting that you can take just that small piece of data and get interesting information like that. So having done all of this molecular study, one of the things that I've been interested in is, could we actually find some molecular changes that might be possible to convert into a simple diagnostic test that every ME patient could go along to where they give blood on the corner of their village or whatever, and it's accessible then to everyone. And the problem with us developing these biomarkers is that often they're not easily examined in a simple way in a chempath laboratory. And so we looked then through our data to see whether we could focus on a molecule which might actually fit this bill of a simple diagnostic test. Rather like you go and have your cholesterol done and you get an HDL and you get an LDL and they give it to you as a ratio. That's the kind of thing that we're looking for. And so, how many? Yeah, okay, good. Yeah, just about. Good. Okay, so we're getting there. So one of the molecules that it's been known about for a time that when there's a chronically activated immune system, changes its molecular state. And this floppy molecule here is a kinase molecule. It's RNA activated protein kinase, or PKR as it's commonly known. So you can see it on the left there. It's very floppy, it's inactive, it's asleep. When it's activated, 2 of the molecules come together, and it's being a kinase, it adds phosphate onto other molecules. But in this case, it phosphorylates itself. So if the immune system is not activated, then the molecule is not phosphorylated. If it's activated, it becomes phosphorylated. So here's a change in the molecular state. So what we've done is to develop antibodies specifically against the phosphate site and purified them, and an antibody against the rest of the molecule So we could actually use this as a potential test. So we're looking for high phospho PKR, that's the phosphate that it's put on itself, and we normalize it against the total PKR molecule. And so we've tested this out just with our cohort of 10 patients in our study. Now those vary from age 12 to 65. They've had ME for various lengths of time, so they're not an ideal study group to actually test it. They're not sort of an acute group that have just, had the severe fatigue for 6 weeks or whatever. And you can see here that it looks quite promising in the sense that the ME patients on the left and lymphocytes, there's certainly an elevation, almost a significance, even with only our 10 patients versus the controls, and similarly in neutrophils, you see a difference not quite as near to significant. So we think this can have promise, but of course would have to be tested out. We'd want to test it out in a homogeneous group of ME patients in terms of how long they've had the illness, and to see how effective it is against patients that are relatively new and are just trying to find out whether in fact they've got the illness. I think actually I will finish there, given that you've been very generous with my time.
We've come a long way, Doctor.
Yeah.
Well, thank you very much indeed.
Yeah.
So, a few questions, please.
Hi, I'd just like to know about what your next steps are with this research, please.
So where will we go?
Yeah. Yes, please.
Okay, so one of the things I've already mentioned is that we are trying to, now we've identified some molecules that change, like, our top 3 transcripts and whatever. We can go to a longitudinal type study, and we won't need squillions of dollars to do that. We can focus on a few of potential biomarkers that have helped sort of explain the nature of the disease, and look to see kind of what happens, say, during the relapse recovery cycle. I mentioned that. But amazingly, and I realised this when we were doing it, when we did our preclinical study, we didn't worry about how long they had the disease, we didn't worry about gender, we didn't worry about age, and that could have completely confounded it. Maybe it was a bit naive, but that's where we started. What we do know when we analysed our data with our principal component analysis, even though the patients stayed within the group, the males and females separated out a bit. And on age 2, we had about 3 that were in the sort of teenage group, and 3 in 30 to 50, and maybe 4 a bit older. And they also they just separated out slightly. So, we'd like to see whether, in fact, if we focused on age and gender and length of illness, whether in fact that actually generates some rich data. Obviously in the diagnostic test I've mentioned, we are sort of trying to head and see whether we can get something which is quite simple and effective, So, but the other thing, could I add just one more, and that is that, the aim of this was to study these patients in a lot of detail, and I've done it in molecular terms. We're now interested in actually extending the studies, for example, looking at their bioenergetic health index using the seahorse analyzer to see whether in fact we can see what their mitochondrial function is like. We're also looking at their epigenetics as well, given that we've got such a bulk of genes that are down-regulated, we want to look at some of the epigenetics.
One last one, sir.
Hi. You mentioned your daughter had her best year when she was pregnant.
Oh, yes, sorry.
Do you think that's an area that should be looked at more in general?
The pregnancy, is that what I heard you say?
Yeah, correct, yeah.
Sorry, it's a bit distorted from up here. As I understand it, a number of women do have improved health when they're pregnant. Not everybody, and as I understand it, some, when they've had their baby and there's dramatic hormonal changes taken, can actually have a relapse. So we certainly, I knew this before, we held our breath with my daughter that she maintained her health pretty well through the first year of the baby's life when she was breastfeeding quite intensively. And then when her natural, I hope she doesn't mind me saying this, but she's a long way away. When her natural I have actually asked her permission that I can talk about her, so I say that. When her natural physiology came back in, she had a pretty horrendous first couple of months. She had a few seizures and things like that, which she used to have, you know, with her acute phase ME. So there was a kind of a dramatic change. Fortunately, that only lasted a couple of months. And her health now, while not as good as it was during the pregnancy or in the first year after birth, is still better, quite a lot better than it was before she became pregnant. So, you know.
Do you think it's an area worth exploring?
I certainly think it is. And I've got 3 women on my research team, and they're really keen that we actually try and recruit a cohort, maybe through Ros Vellings' patient, you know, woman and encourage them to get pregnant so we can actually do the studies. They said that, not me. It's quite interesting that, you know, sort of wry comment I have to myself is when your daughters are young, you just hope they won't go and get pregnant, and then you get to a stage, you're just hoping they will get pregnant.
You might get support from some organisations if you ran a baby bureau, you know. But I wouldn't trust it, you know.
No.
Thank you very much,
Good.
Warren. Thank
Thank
you.
you.